Originally, section 25.4 was supposed to be with 25.5. We didn't get them put in the same video, so this will be the lecture for section 25.5, the last section of chapter 25, and it's going to cover gene mutations. We've already looked at a few different types of gene mutations in the last chapter, but let's look at what specifically happens during a mutation. A mutation, by definition, is a permanent change in the sequence of bases in the DNA. This effect can range from no effect at all to a complete inactivity of the protein that's being coded for. A germline mutation can be passed on to subsequent generations, and some mutations can lead to cancer. There are three causes of mutations. There can either be an error in replication. Proofreading by DNA polymerase usually takes care of it, so DNA polymerases are spell check when it comes to replication. Mutagens, which are things that can cause mutations to occur, such as radiation and certain organic chemicals, can be a cause of a mutation. The DNA repair enzymes constantly monitor and repair any irregularities. Sometimes they don't repair them the way that they should and don't get repaired because of so many different areas of damage that are done due to radiation and organic chemicals and other things called mutagens. We also have transposons, which are jumping genes. These are specific DNA sequences that move within and between chromosomes and can cause a problem. Sometimes they alter neighboring gene expression as well. Likely all organisms, including humans, have transposons. Here's an example of a transposon. So we have a normal gene that codes for purple pigment. We have a mutated gene, and this is a transposon, a section of DNA that wasn't normally there, and it codes for no pigment instead of purple pigment, so you see it show up like this in corn. Look at the effect of mutations on protein activity. Point mutations involve a change in a single DNA nucleotide. Possible change in a specific amino acid when this occurs may have no effect at all. It just depends on what gets changed. It may produce an abnormal protein. An example of a point mutation that causes an illness is sickle cell anemia. It also may cause it to produce an incomplete protein. So some of you on the virtual lab experienced this type of a mutation. So if you notice that this section of DNA has no mutation in it, and it's going to code for this particular set of amino acids to form a protein. This is a mutation that's a point mutation here. So we have, instead of a G, we have an A instead. And if you notice, you don't see any difference at all because it codes for the same protein, because we know that there are different triplets that code for particular proteins or particular amino acids. So this one didn't make any difference that that was a mutation there with a point mutation. Here's an example of a point mutation, though, in the middle of this one that caused a change in the amino acid sequence. So instead of proline, glutamine, glutamine, we end up with proline, valine, glutamine. And here's an example where a point mutation caused a triplet to code for the stop amino acid or the stop codon on the messenger RNA. So it stopped or is an incomplete protein. So we have a normal protein with a point mutation. We have an abnormal protein with a point mutation. And we have an incomplete protein because of a point mutation. A frame shift mutation is the next type we're going to look at. This is when you have one or more nucleotides that either get inserted or deleted from a section of DNA. The result can be a complete new sequence of codons and the whole protein being non-functional. Let's look at an example of that. So we have the sentence, the cat ate the rat. So what happens when we delete the C out of the sequence? It becomes nonsense at that point. The atta, tet, her, at. That has nothing to do with the cat ate the rat. So it becomes nonsense or non-functioning protein instead. Non-functional proteins. A single non-functioning protein can have a dramatic effect on the phenotype. Remember, phenotype is the physical appearance of an organism because enzymes are often a part of a metabolic pathway. An example of this is PKU syndrome or phenylketonuria. Phenylalanine cannot be broken down and it builds up, causing mental impairment. Another example is androgen insensitivity. This is a faulty receptor that makes cells unable to respond. The genetic male looks like a normal female, but genetically is a male instead. Mutations also can cause cancer. In the United States, at the time of the production of this book, the three deadliest forms of cancer were lung, colon and rectal cancer, and breast cancer. The development of cancer involves a series of accumulating mutations that can be different for each type of cancer. Carcinogenesis, or the beginning of cancer, begins with the loss of the tumor suppressor gene activity and or the gain of oncogene activity. So this is what happens when you have a mutated tumor suppressor gene. A mutated tumor suppressor gene codes for a product that directly or indirectly no longer inhibits the cell cycle, so the cell grows out of control. In this figure, we see an example of one that deals with an oncogene. So an oncogene codes for a product that either directly or indirectly overstimulates the cell cycle, causing uncontrolled growth of the cell. Although cancers vary greatly, they usually follow a common multi-step progression. Most cancers begin as abnormal cell growth, and it's called benign, and benign means not cancerous, and it usually doesn't get much larger than what it is. Growth may become malignant, though, meaning that it can become cancerous and possess the ability to spread. Here you notice we have a cell that's dark pink. It acquires a mutation for repeated cell division, and this is in skin cells, epithelial cells of the skin. The new mutations arise, and one cell, it's now green, has the ability to start a tumor. The tumor is at its place of origin, right there at the beginning, and it reproduces and reproduces and reproduces. And if you notice down here, we have a blood vessel and a lymphatic vessel. The cells gain the ability to invade underlying tissue, so it's not any longer just an epithelial issue. It goes down below the epithelium and produces a proteinase enzyme. And this cancer cells now have the ability to invade lymphatic and blood vessels. And the blood vessels, of course, are your circulatory system. Your lymphatic vessels are the ones that help with your infection fighting. And this travels all throughout your body. And because that travels all throughout your body, new metastatic tumors are found some distance from the original tumor, so it's spread throughout the body. This just shows a more step-by-step what's going on in each one of those pictures. Characteristics of cancer cells. Cancer cells are generally genetically unstable. They don't correctly regulate the cell cycle, which causes them to reproduce out of control. Cancer cells escape the signals for cell death, so even though the cell may say it's time for apoptosis to occur or cell death to occur, it doesn't catch that signal and continues to reproduce. The cancer cells can survive then and proliferate or grow elsewhere in the body, traveling via the blood system or the lymphatic system, and then show up in a different organ and cause cancer in other organs as well. The good news is we are starting to catch cancer, all different types of cancer, at an earlier stage because we have diagnostic tests that help us to look for those different types of cancer that are there. We also are much more careful about family history, looking at that, and being able to identify the carcinogens, because now we know, like they didn't know back in the 40s and 50s, that smoking cigarettes is bad for your health because it causes cancer. So we've learned these things as we go along and try to stop those behaviors and the partaking of those things that can cause the cancer to even start in the first place. So diet, exercise, and checking family history are the three things that are going to help us to prevent cancer in us. And as you all get older, you're going to find that they're going to have earlier and earlier detection for the different types of cancers as well.